Hello everyone, my name is P. Nandakumar. I have come from Telangana Tribal Welfare Residential Degree College, Main Kamareddy. The topic that I have chosen for today is RDNA technology from the subject of zoology. Presently, I am pursuing in degree final year. So friends, before getting into the topic, I want to ask you one question that, can we change the fate of the organisms? No, we can't change the fate of the organisms because it is not there in our hands. If you observe the surroundings, there are many plants and animals which are suffering from many diseases and genetical disorders, right? So it is their genetic fate to having those diseases. But what I want to say is, we can change the genetic fate of the organisms. But how? Is it possible? Yeah, it's possible. It is possible through a technology called as RDNA technology. So this RDNA technology allows us to change the genetic fate of the organisms. So with this RDNA technology, we can directly intervene into the genetic fate of the organisms. So if we had an intent to change the organisms into a better way, so this RDNA technology will very helpful to us to change that. So what is this RDNA technology? If you observe the word RDNA technology, it stands for recombinant DNA technology. Here there is a word recombination in it. So here, what is recombination? Recombination is nothing but the combination of two different genetic material to form a new genetic material is called as recombination or the genetic recombination. So you may have a doubt like where this recombination is occurring. This recombination is occurring in the nature. So in the nature, during the gametogenesis in the meiosis phase, the genetic recombination is occurring with the crossing over of the chromosomes, and if the male and female gametes gonna fuse to form a new genetic material, that is progeny material, which is a quite variant material from the parental DNA. So this is the genetic recombination that we have. So this is the natural evidence that we have about the genetic recombination. We have enriched data from the biological sciences like cell biology, molecular biology, genetics and biochemistry. With this enriched data, we can create artificially the genetic recombination in the laboratories. This is nothing but the RDNA technology. So it is simply an art of cutting and pasting of genes of our interest to produce our desired product. So coming to the definition of this RDNA technology, so it is defined as the study and manipulation of DNA to achieve a desired goal in a pre-decided way is called as RDNA technology. Once again I will say, this is the study and manipulation of DNA to achieve a desired goal in a predetermined way is called as RDNA technology. So this technique allows us to change the genetic fate of the organisms. So with this technology, we can directly intervene into the genetic fate of the organisms. So this technology was emerged. This technology was emerged after the discovery of restriction endonucleases by a scientist namely W. Arbor in 1962. So this technology got possible after the discovery of restriction endonucleases. So after that, two scientists namely Cohen and Boyer, who are the scientists, discovered they made successfully recombinant DNA in their laboratory, and they are the discoverers of this RDNA technology. So what we will do in this RDNA technology means, we will take our desired DNA and we will put into another DNA to form a recombinant DNA, and that is introduced into the host cell like bacteria, which is cultured to get the desired products. So see the picture, which is titled with insulin hormone production. So insulin is the first therapeutic product which is produced by the application of recombinant DNA technology. So what we will do in this means, we will take the insulin gene to produce the insulin hormone. So insulin hormone production is actually a biological process in which the beta cells of the pancreas produce the insulin hormone. But we will take the insulin gene from the pancreas and we will put into another gene to form a recombinant DNA, and that is introduced into another DNA to form a recombinant DNA, and that is introduced into the host cell to produce the insulin hormone. So such clones are capable of producing fair amount of insulin hormone. So nowadays there are many people who are suffering from diabetes mellitus, right? So if we give this artificially produced insulin to them, then the disease can be rectified. So what is the process of this RDNA technology, and what are the steps involved in this RDNA technology? Means there are mainly 6 steps involved in this RDNA technology. They are nothing but, first one is selection and isolation of desired DNA and selection of suitable cloning vector, and third one is production of recombinant DNA, and the fourth one is introduction of recombinant DNA into the host cell, and the fifth one is selection of transformed cells, and the sixth one is expression and multiplication of desired DNA in the host cell. So these are the steps that are involved in this RDNA technology. So coming to the first step, that is selection and isolation of desired DNA. So first we have to identify the cell which is having our desired DNA. Here, what is desired DNA? Desired DNA is nothing but the piece of DNA which is containing our gene of interest is called as desired DNA. So now we have to extract that desired DNA. In order to do that, first what we have to do means we have to treat the cells with the lysozyme and the detergents to break down the cell wall and the cell membrane of the cells. After that, we have to treat the cells with the phenol, proteases and the ribonucleases. After that, what we have to do means the cells are subjected with the ultracentrifugation for the purification of the DNA. Then we will get the supernatant as in the DNA molecule, supernatant in the DNA. So what we will do this DNA molecule means, now we have to add restriction endonucleases to the DNA molecule to get the desired product fragment. So these fragments are having 2 sticky ends at the both sides. So what is the help of these sticky ends means, these are the single strands which will help us to attach with the another DNA. So these DNA fragments are separated by a technique called as gel electrophoresis, and these DNA molecules are identified by a technique called as Southern blotting technique. So this is the first step that is involved in this RDNA technology. So see the picture, a cell which is containing our desired DNA. So in this way, we will take our desired DNA from the cell and we will put into another DNA. So coming to the second step, that is selection of suitable cloning vector. Here, what is vector? Vector is nothing but it is a vehicle to carry our desired DNA. So vehicle is actually a self-replicable molecule. So generally we prefer to use prokaryotic vectors because they have rapid multiplication capacity. So coming to the plasmid as a vector, which is a bacterial vector. Now what is plasmid? Plasmid is a extra chromosomal double stranded circular DNA which is present in the bacteria. Now we have to extract that plasmid. So how we will extract that plasmid means, now we have to treat the bacterial cells with the enzyme EDTA and the lysozymes to break down the cell wall of the bacteria. After that, we have to do ultracentrifugation for the purification of the plasmid. Then we will get the plasmids. So now we have to use again the restriction of the same restriction enzymes to get the sticky ends of the plasmid. So coming to the third step, that is production of recombinant DNA. So what is here recombinant DNA means? It is having the two genetic material. So in this step, what we will do means we will combine our desired DNA with the plasmid DNA to form a recombinant DNA. So this process is also called as ligation. So for this ligation, an enzyme is required that is called as DNA ligase. So with this DNA ligase, we can attach the 2 DNA fragments. So for this ligation, an ATP molecule is required for the formation of phosphodiester linkages or the covalent bonds between the 2 DNA strands. So this is the process, that production of the recombinant DNA. So coming to the fourth step, that is introduction of recombinant DNA into the host cell. Here what is the purpose of introduction of our recombinant DNA into the host cell means to multiply our desired DNA to get the fair amount of the product. So there are many methods to introduce our desired DNA into the host cell. So some of them are transformation, transduction, transfection and electrophoration and microinjection. So these are some methods which we will introduce our recombinant DNA into the host cell. So see the picture, now our recombinant DNA is introduced into the bacteria. So coming to the fifth step, that is selection of transformer cells. Here in the bacterial culture, the 2 cells become 4 and 4 to 8 and 8 to 16 and so on. So during the transformation, some of the cells do not uptake our desired DNA or the recombinant DNA. So those cells do not contain the recombinant DNA are called as non-transformer cells, and those cells which are containing our desired DNA are called as transformer cells. Now we have to separate the transformer cells from the non-transformer cells. So what we will do? We will do antibiotic resistance tests. So in the culture we will use the antibiotics like ampicillin, tetracycline. So with this usage, the only transformer cells which are containing the antibiotic resistance gene in the plasmid, so they will only survive, and the non-transformer cells will be died. So see the picture, there are both transformer cells and the non-transformer cells, but only after the addition of this antibiotics, only the transformer cells are alive and the non-transformer cells are died. So this is a fifth step that is involved in this RDNA technology. So coming to the sixth step, that is expression and multiplication of desired DNA in the host. So we selected the transformer cells, right? So transformer cells are containing our desired DNA, so that is expressed in the host cells. For example, if we use the insulin hormone gene and that is introduced into another DNA to form a recombinant DNA, and that is introduced into the host cell to form the insulin hormone as a product. So the transformer cells are multiplied to obtain the sufficient number of copies. So these are the 6 steps that are involved in this RDNA technology. So take an overall view. First we have to identify the cell having our desired DNA, and we have to use restriction endonuclease for the extraction of our desired fragment, and we have to also take the plasmid from the bacteria, and we have to again use restriction endonuclease which are the same, and we have to get the both sticky ends from them. So now we have to add DNA ligase to get the recombinant DNA. Now that recombinant DNA is introduced into the bacterial culture to produce the number of copies of our desired product, and our desired product can be gained by this process of RDNA technology. So see the picture, that is human cell containing the desired DNA, and we have also taken the plasmid from the bacteria, and we both fuse it by recombinant DNA technology. So what are the tools involved in this RDNA technology? So coming to the tools, they are mainly enzymes and host cell and the vector, and some of the other tools are linkers and adapters. So coming to the enzymes, so enzymes are classified mainly into 2 types. They are exonucleases and the endonucleases. So coming to the exonucleases, these are the enzymes which cuts the DNA molecule in the outer nucleotides. Coming to the endonucleases, these are the enzymes which cuts the DNA molecule in the outer nucleotides. So coming to the main enzymes, that are restriction endonucleases. So restriction endonucleases are mainly 3 types. They are restriction endonuclease 1 and 2 and 3. So restriction endonuclease 1 and 3 are not important for us in this RDNA technology because they cut the DNA molecule out of the recognition sites, but restriction endonuclease 2 is important for us because it cuts the DNA in the recognition sites. So now we will take the restriction endonuclease 2 for our RDNA technology. So coming to another important enzyme, that is DNA ligase. So DNA ligase is also called as molecular glue, in which it attaches the 2 fragments of the DNA. It requires an ATP for its function to form the phosphodiester linkages or the covalent bonds between the 2 DNA strands. So our restriction endonucleases are also called as molecular scissors. So coming to the another tool, that is the cloning vector. Here cloning vector is the vehicle to carry our desired DNA. So generally we prefer prokaryotic vectors because they have rapid multiplication capacity. And there are another vectors also used, namely cosmid, bacteriophage, and bacterial artificial chromosome, and yeast artificial chromosome, and mammalian artificial chromosomes. So these are the another vectors used to carry our desired DNA. So coming to the host cell, there are mostly prokaryotic vectors also used and the eukaryotic vectors also used. So in these cells, so prokaryotic vectors includes the E. coli. If apart from the E. coli, there are also Plasmodium, and Salmonella. So these are also used. So coming to the eukaryotic vectors, the yeast cells and mammalian ovarian cells are also taken. So another tools like the linkers and adapters also used to produce the cohesive ends in the DNA ligase, in the DNA strands. So these are some tools which are required for this rDNA technology. So coming to the applications of this rDNA technology, there are many applications are there through this rDNA technology for the human welfare. So first one is the production of transgenic animals. So we will produce the transgenic animals like transgenic cow and transgenic sheep. Here transgenic sheep can produce by inducing the growth hormone. So it can produce the more number of meat for us, and it can give the more number of wool also. So coming to the transgenic cow, it produces the more number of protein like casein, and also it gives more number of milk for us. So coming to the transgenic plants, so there are plants like BT cotton and BT brinjal, which are the resistance to insects and insecticides. So these are the another application. So coming to the production of hormones, there are some hormones like insulin also produced through this rDNA technology, and production of vaccines also. And this rDNA technology is also used in the application of in the forensic sciences like DNA fingerprinting, and it is also used in the production of biofuel like biohydrogen, biomethanol, bioethanol. So these are some applications. So recombinant DNA is also helpful in the production of therapeutic products like vaccines, growth hormones, and antibodies, vectors, and recombinant proteins, and anti-cancer drugs. And these genetically modified products like fruits and genetically modified vegetables, crops, and microbes and animals also, genetically modified crops also produced. So coming to the diagnosis, so these are also used in gene therapy, and CRISPR, which is used in the elimination of diseased genes by the use of Cas-protein 9, and also used in the monitoring devices and therapeutic strategies. And another energy production applications like production of biohydrogen, bioethanol, biomethanol, and the biobutanol. So these are the applications which are used rDNA technology. So we will recall our classes again. So coming to the definition, this is the study and manipulation of DNA to achieve a desired goal in a predetermined way is called as rDNA technology. So this technology allows us to change the genetic fate of the organism. We can produce the hormones like insulin also through this technology. We have to introduce the insulin gene into the another DNA to form a recombinant DNA, and that is introduced into bacterial culture to produce the insulin hormone. So these are the steps, 6 steps that are involved in this rDNA technology. So first we have to select the cell, and we have to isolate the desired DNA, and after that we have to fuse with the cloning vector DNA to form a recombinant DNA, and that is introduced into the bacterial culture to produce our desired product in a fair amount. So thank you for giving this golden opportunity to our Honorable Secretary Sir Ronaldo Sir Garu, and my principal Annapurna Madam Garu, and my mentor teacher Manohar Sir Garu, and thank you everyone.